Yeah, hello, my name is Bright Ocansey, a doctoral researcher with the Korle-Bu Teaching Hospital in Accra and the University of Manchester. Today, I'll be talking to you about the epidemiology and diagnosis of African histoplasmosis. The learning objectives of this presentation are to get an overview of histoplasmosis in humans, to get an insight into the epidemiology of African histoplasmosis, and to be familiar with the clinical manifestations of African histoplasmosis, and finally, to appreciate the various ways of making a diagnosis of African histoplasmosis. Histoplasmosis is caused by the thermally dimorphic fungi, Histoplasma capsulactum. Historically, there are three variants. That is variant capsulactum, duboisii, and fasciminosum. Capsulactum and duboisii are known to cause human infections, whilst the variant fasciminosum causes infection in horses. An infection with variant capsulactum is known as classical histoplasmosis because that was the first form that was described by Samuel Darling in 1906. It is sometimes also known as American histoplasmosis because of its established endemicity in the Americas, that's the Northern, Central, and then South America. An infection with var duboisii, on the other hand, is commonly referred to as African histoplasmosis because almost all cases that have been reported are from Africa. However, this historical classification is challenged by current phylogenetic studies that suggest that the genus Histoplasma may be more diverse and there may be as many as 16 phylogenetic species. This includes the African phylogenetic species, which encompasses almost all strains of duboisii and some strains of capsulactum and fasciminosum. The various advances that has been made in classical histoplasmosis are yet to be replicated in African histoplasmosis, such as the epidemiology, pathogenesis, diagnosis, and management. So in terms of epidemiology in the US and then other parts of America, we have some clear indication of where the hotspots are. And so they were able to establish surveillance programs and also make it a notified case where cases are being notified with authorities. And the pathogenesis is quite clear as well. In terms of diagnosis, most of these new non-culture-based assays that are being made are made with isolates of Histoplasma capsulatum, the variant capsulatum. And there are established guidelines available for the management of classical histoplasmosis as well. So in terms of epidemiology, clinical features and the laboratory characteristics, they are different from that of, they differ in terms of the two forms of histoplasmosis in humans. So with the classical histoplasmosis, even though it's more common in America, it has a global patchy distribution. And so it's also quite often seen in the Southeast Asia and also common in Africa as well. The pathogenesis is quite clear. Inhalation is the infection routes and tends to affect lungs more, and sometimes bone marrow, and then other auxiliary sites includes the skin and then central nervous system. It's mostly associated with immunosuppression, mostly HIV, and then solid organ or stem cell transplants. In the lab, they tend to have a smaller size between 3 to 5 micrometers, and their cell walls are thin. Whilst African histoplasmosis is more restricted to Africa, especially in the central and the western parts. The pathogenesis is not quite clear. Inhalation, there will be several postulates. The popular one is inhalation, similar to that of classical histoplasmosis. There's been some transcutaneous ones that have been suspected as well. It commonly affects the skin, the lymph nodes, and then the bones, and rarely affects other parts, say the lung, even though there are occasional case reports of such. And it's rarely associated with immunosuppression. Most cases are diagnosed in immunocompetent people, even though cases in HIV and then other immunosuppressed patients have been reported. In the lab, the cell size is bigger than that of variant capsuloptum, so between 7 to 15 micrometer, and the cell was Accra 2. So the first case of African histoplasmosis was described by James Duncan whilst in 1943 while studying a case that has originally come from Ghana in West Africa. So whilst seeing the case, he realized the E source in this tissue was larger than that reported or first described by Samuel Darling, and so thought it was a large form of histoplasmosis. So later, Belgium scientist Raymond van Bruggen studied more of that cases that later was diagnosed in other parts of Africa. Received several isolates from Albert du Bois, and then in 1952, he concluded that it would be a variant of the original capsuloptum, and then named it after Albert du Bois. So it's very much restricted to Africa, and then almost all cases, even diagnosed outside the continent, have a travel history to Africa. People that travel to Africa or stayed briefly or for a long time in Africa have happened to present with a case of African histoplasmosis. However, there's one indigenous case from India. There's someone that hasn't traveled outside India before as well. It tends to affect apparently healthy individuals, like I've previously mentioned. African histoplasmosis is relatively rare, but it is believed that several infections are probably missed due to inadequate awareness and the fact that in endemic areas, access to histopathology and other mycology services for diagnosis is very poor. And then about close to 90% of cases are from the western and central part of Africa. So far, approximately 500 cases have been reported from 32 African countries, and 30% of them diagnosis was made outside Africa. So these are African indigenes that migrate to the West, and then they tend to develop African histoplasmosis, and then diagnosis was made in the West. There have been two large studies. These are single center experience retrospective studies from DR Congo and Togo. In DR Congo, they reported 36, so this was a single facility. They reported 36 cases over 6 years, whilst in Togo it was 17 over a period of 15 years. So the current, distribution of cases and mapping really relies on this sporadic, reported cases, and then occasionally or rarely on serology and then histoplasma skin testing as well. So this beautiful map was by Oladele and colleagues, depicting or showing the 32 African countries. So here you could see the western part where you have more of the variant du Bois, and then down in the south you see more of the capsuloptum variants as well. Histoplasma skin sensitivity testing is also very instrumental and has been used in classical histoplasmosis in the US and other parts of the Americas to define the exact hotspots. And in Africa, this has been used as well. And then rate has been reported as high as 35%. So this was around Bat Cave, where previously the variants du Bois was cultured from. But because the histoplasmin that is used for this sensitivity testing is prepared from the variant capsuloptum, and it tends to cross-react with that of the variant du Bois. When you do the test, you are not really sure. When you do the survey, you are not really sure which one is captured. So, and we have both, quite common in Africa too. So it's really a problem to define exactly the epidemiology or the hotspots in Africa using this approach. In terms of the disease, the males tend to be affected more, the male to female ratio of averaging 2:1. But all age groups may be affected, and cases has been reported in people as young as 13 months and then as old as 70 years. And in terms of the case series and reviews, the median age range has been between 21 to 47 years. And most cases have been associated with, agricultural activities, and then collecting of bed and then bat guano, as well as living in, bat infested, homes, but not for all cases, because there are some cases that people are not having any link with any of these epidemiological risk, factors. The pathogenesis is not really studied, and then the current data is scanty. But it's believed to be share similar infection rate with that of classical histoplasmosis. So the microcanidia is inhaled and then subsequently spread via hepatogenous and then lymphatic spread to the lymph and then the skin and then bones, where the lesions then tend to develop. But various, forms have been considered for transcutaneous routes after trauma through acupuncture sites, tribal marks, tattoos, and then insect bites. The incubation period is not known, and then cases has been diagnosed weeks and then several years, as long as 40 years after presumed, exposure. So it's difficult to define a proper incubation period for African histoplasmosis. Appearance of lesions may appear as single lesions on a part of body, or you have sometimes scanty lesions confined in a particular area, as you can see in A and B respectively. And mostly these ones do occur with no systemic involvement, or you may have multiple skin lesions all over the body, mostly on the face and then on the trunk, sometimes at the back. And in disseminated cases, they tend to affect bones more, and then sometimes also internal organs. These lesions may be papular, they may be nodular, they may appear ulcerative, eczematoid, or psoriasiform as well. The lesions may appear in different sizes, because whilst the old ones tends to heal, the new ones appear. So you realize they have different sizes of the lesions on the skin. The smaller lesions may resolve without suppuration, whilst larger lesions may develop into soft cold abscess, as you could see. You can see here in the figure E here. Skin lesions are usually accompanied by lymphadenopathy or lymphadenitis, and they are mostly superficial, confined to an area of lymph drainage, or maybe generalized when the lesions are disseminated. Bones are commonly affected, and it affects about 30%. In 30% of cases, bones are involved, and it tends to affect more of the extremities. So the femur, the humerus, the tibia, wrists are mostly affected, and then the vertebra as well. In rare cases, internal organs are affected, and they tend to mimic cancer a lot. So in most cases, they are not suspected, but they are accidentally detected in the lab. So to arrive or establish a clinical suspicion of African histoplasmosis is not straightforward, because there are several differential diagnoses in terms of all forms of appearance, cutaneous, subcutaneous, or osseous manifestations. This even includes some endemic and then systemic, fungal infections such as sporotrichosis, cryptococcosis, megomycosis, chromoblastomycosis, mycetoma, and then blastomycosis as well. There are various laboratory approaches for diagnosing African histoplasmosis. Then direct microscopy happens to the simplest of them, or so basically the approach or how to prepare your sample depends on the available reagents. So you could use the potassium hydroxide reagents, you could use the Jameson stain, lactophenol cotton blue stain, or the silver stain. You could use gram stains, but in most cases they appear to be negative, so it may not be very helpful. So on microscopic examination, you will see a thick wall, oval, and double contoured cell with narrow burden. They may be in isolation or in chains, and in rare cases, clusters. And the yeast cells show this typical hourglass or figure 8 burden. And sometimes you see bad scars too. So these are the various using different things and then reagents for the diagnosis. So it depends on what the lab have. So it means with the little that we have, even with QH, you should be able to at least make a preliminary diagnosis of African histoplasmosis in your lab. Culture is very important, and then until that evolution of molecular techniques was the only means that could be used to differentiate between the two variants that causes human histoplasmosis. But culture is very slow and may take up to 8 weeks. It also requires at least biological safety level 2, so as to prevent laboratory acquired infection whilst manipulating the culture, and not from the clinical specimen per se. That's if you follow the required good clinical microbiology protocols. So various samples can be used, including skin biopsy or biopsy from the lymph nodes as well. And then pus or any suppurative material. So because it's dimorphic, you could grow it at room temperature and then at 37 as well. So at room temperature, you could use sabrodestrous agar, and then at 37, you use a brain heart infusion. So at room temperature, the forward coloring morphology shows white-brown coloration with a fine, dense cottony texture. The reverse is mostly white, but can be yellow as well. And microscopy initially shows numerous small, smooth-walled microconidia measuring between 2 to 4 micrometers. But as incubation goes, as for African histoplasmosis. And then mostly it's important to use the fungal stains, so silver stain or periodic as a chef, so that it demonstrates the yeast cells proper. So on microscopic examination, you're likely to see aggregates of multinucleate germ cells containing the oval double counted E cells. In the tissue too, they exhibit, same, hourglass or figure 8 pattern. And then you may also see them in isolation, or in chains, and then occasionally in clusters. The most common tissue reaction is the demonstration of the granulomatous reaction, and with some acute inflammation response, as well. The organisms are mostly, numerous in infected tissue and then scanty in healing fibrotic lesions. Experience is needed to avoid, confusing the C cells with that of the variant capsulatum, or even capsule deficient, Cryptococcus species, as well as small variants of Blastomyces species. But you could use a positive reaction of the MISI coming to separate, Cryptococcus species. And then you have to carefully look at the single, broad base in the small variant of Blastomyces species, as well as their multinucleation as well. So there are non-culture based assays, antibody, antigen assays, available for histoplasmosis, but they've been rarely used in terms of African histoplasmosis. Quite common and more often used in classical histoplasmosis. But some immunodiffusion and then immunofluorescence techniques have been employed in African histoplasmosis, but they seem to lack consistency, and then, as usual, are not able to differentiate between the two, variants. The enzyme immunoassays and then immunochromatography tests too are available. They appear to be more consistent and then are very helpful. And the available commercial histoplasma kits currently available both for EIA and then ICT are believed to detect variant WBC antigens as well, even though there are no evaluations that is to that effect. Molecular techniques appears to be, one of the ways you could distinguish between the two, in addition to let's say, using MALDI-TOF. You could do that on fresh samples. There was a case that diagnosis was made within 24 hours using molecular techniques because suspicion was very high. Or you could work on archived tissue blocks as well to make the diagnosis as well. So it may typically involve doing either a pan-fungal PCR or doing a histoplasma-specific PCR and then following that up with a PCR sequencing, the DNA sequencing, of the PCR products. Imaging is very important, especially when bones are involved, and may be used for diagnosis, to identify dissemination and to monitor, therapy or identify relapse. The various modalities of imaging have been used and previously described in cases of African histoplasmosis and have been documented to be very useful. Even though these findings are not specific to African histoplasmosis, some of the very common ones that have been associated with it are listed here. And it's important to note that a reduction in the medullary differentiation of distal metaphysis is one of the early signs of the involvement of bones, and it must be looked out for. So in summary, African histoplasmosis is an indolent infection caused by Histoplasma capsulatum variant WBC, much common in the west and then the central part of Africa. The pathogenesis is not clear, but the route of infection is believed to be similar to that of classical histoplasmosis. It differs from the clinical manifestations, epidemiology, as well as the laboratory characteristics from that of classical histoplasmosis, even though they share some similarities, such as having the same mycelia phase, and it appears to affect people that are apparently okay, and the appearance of Skin and then lymph nodes and bulges are the common manifestations. Diagnosis is not straightforward, and then it has several differential diagnoses. This includes even endemic and then systemic fungal infections as well. Conventional laboratory methods still play a role, and they are very helpful, especially in settings that don't have access to advanced techniques. But bear in mind, for you to be sure about the etiology, it may require molecular techniques or MADITOF. Imaging is important, and then especially in diagnosis and prognosis, especially when bones are involved, and the various modalities have all been previously used and then described. Thank you.